One of the first mutants Morgan observed turned out to be one of the most instructive. While normal fruit flies have red eyes, these had white ones, and he noticed that the white-eyed flies were typically male. It was known that the sex of a fruit fly, or for that matter the sex of a human, is determined chromosomally. Females have 2 copies of the X chromosome, whereas males have 1 copy of the X and 1 copy of the much smaller Y. In light of this information, the white eye result suddenly made sense. The eye color gene is located on the X chromosome, and the white eye mutation, W, is recessive. Because males have only a single X chromosome, even recessive genes, in the absence of a dominant counterpart to suppress them, are automatically expressed. White-eyed females were relatively rare because they typically had only 1 copy of W, so they expressed the dominant red eye color. By correlating a gene, the one for eye color, with the chromosome, the X, Morgan, despite his initial reservations, had effectively proved the Sutton-Boveri theory. He had also found an example of sex linkage, in which a particular characteristic is disproportionately represented in one sex. Like Morgan's fruit flies, Queen Victoria provides a famous example of sex linkage. On one of her X chromosomes, she had a mutated gene for hemophilia, the bleeding disease, in whose victims proper blood clotting fails to occur. Because her other copy was normal and the hemophilia gene is recessive, she herself did not have the disease, but she was a carrier. Her daughters did not have the disease either. Evidently, each possessed at least 1 copy of the normal version. But Victoria's sons were not all so lucky. Like all males, fruit fly males included, each had only 1 X chromosome. This was necessarily derived from Victoria. A Y chromosome could not have come only from Prince Albert, Victoria's husband. Because Victoria had 1 mutated copy and 1 normal copy, each of her sons had a 50-50 chance of having the disease. Prince Leopold drew the short straw. He developed hemophilia and died at 31, bleeding to death after a minor fall. 2 of Victoria's daughters, Princesses Alice and Beatrice, were carriers, having inherited the mutated gene from their mother. They each produced carrier daughters and sons with hemophilia. Alice's grandson, Alexis, heir to the Russian throne, had hemophilia, and would doubtless have died young had the Bolsheviks not gotten to him first. Morgan's fruit flies had other secrets to reveal. In the course of studying genes located on the same chromosome, Morgan and his students found that chromosomes actually break apart and reform during the production of sperm and egg cells. This meant that Morgan's original objections to the Sutton-Boveri theory were unwarranted. The breaking and reforming, recombination, in modern genetic parlance, shuffles gene copies between members of a chromosome pair. Now, this means that, say, the copy of chromosome 12 I got from my mother, the other, of course, comes from my father, is in fact a mix of my mother's 2 copies of chromosome 12, one of which came from her mother and one from her father. Her 2 12s recombined, exchanged material during the production of the egg cell that eventually turned into me. Thus, my maternally derived chromosome 12 can be viewed as a mosaic of my grandparents' 12s. Of course, my mother's maternally derived 12 was itself a mosaic of her grandparents' 12s, and so on. Recombination permitted Morgan and his students to map out the positions of particular genes along a given chromosome. Recombination involves breaking and reforming chromosomes. Because genes are arranged like beads along a chromosome string, a break is statistically much more likely to occur between 2 genes that are far apart, with more potential breakpoints intervening on the chromosome, than between 2 genes that are close together. If, therefore, we see a lot of reshuffling from any 2 genes on a single chromosome, we can conclude that they are a long way apart. The rarer the reshuffling, the closer the genes likely are. This basic and immensely powerful principle underlies all of genetic mapping. One of the primary tools of scientists involved in the Human Genome Project and of researchers at the forefront of the battle against genetic disease was thus developed all those years ago in the filthy, cluttered Columbia fly room. Each new headline in the science section of the newspaper these days along the lines of "Gene for something located" is a tribute to the pioneering work of Morgan and his boys. The rediscovery of Mendel's work and the breakthroughs that followed it sparked a surge of interest in the social significance of genetics. While scientists had been grappling with the precise mechanisms of heredity through the 18th and 19th centuries, public concern had been mounting about the burden placed on society by what came to be called the degenerate classes, the inhabitants of poorhouses, workhouses, and insane asylums. What could be done with these people? It remained a matter of controversy whether they should be treated charitably, which the less charitably inclined claimed ensured such folks would never exert themselves and would therefore remain forever dependent on the largesse of the state or of private institutions, or whether they should be simply ignored, which, according to the charitably inclined, would result only in perpetuating the inability of the unfortunate to extricate themselves from their blighted circumstances. The publication of Darwin's Origin of Species in 1859 brought these issues into sharp focus. Although Darwin carefully omitted to mention human evolution, fearing that to do so would only further inflame an already raging controversy, it required no great leap of imagination to apply his idea of natural selection to humans. Natural selection is the force that determines the fate of all genetic variations in nature. Mutations like the one Morgan found in the fruit fly eye color gene, but also perhaps differences in the abilities of human individuals to fend for themselves. Natural populations have an enormous reproductive potential. Take fruit flies, with their generation time of just 10 days, and females that produce some 300 eggs apiece, half of which will be female. Starting with a single fruit fly couple, after a month, i.e. 3 generations later, you will have 150 times 150 times 150 fruit flies on your hands. That's more than 3 million flies, all of them derived from just one pair in just 1 month. Darwin made the point by choosing a species from the other end of the reproductive spectrum. The elephant is reckoned to be the slowest breeder of all known animals, and I have taken some pains to estimate its probable minimum rate of natural increase. It will be under the mark to assume that it breeds when 30 years old and goes on breeding till 90 years old, bringing forth 3 pairs of young in this interval. If this be so, at the end of the 5th century there would be alive 15 million elephants descended from the first pair.